Right. Good morning, everyone. I want to give a very warm welcome to Ryan Corcoran today, our speaker for the Frontiers in Oncology seminar series. Ryan is on the faculty at Harvard Medical School and director of the GI Cancer Program, as well as the Termeer Center for Targeted Therapy at Massachusetts General Hospital Cancer Center. Ryan received his undergraduate degree from Princeton University, where his research career started in Arnie Levine's lab working on oncogenic Wnt signaling. Ryan then came to Stanford here to do his MD PhD. And during his graduate work with Matt Scott, where I first got to know Ryan, he did really beautiful work on understanding how cholesterol metabolites play a key role in regulating hedgehog signaling in medulloblastoma. And this work ended up having a really important impact on the hedgehog signaling field and continues to really drive a lot of research in this area. After that, Ryan moved to Boston, where he completed his residency and fellowship at Mass General Hospital and the Dana-Farber Cancer Institute. And over the last decade, Ryan has really become a leader in delivering on the promise of personalized cancer medicine. He's worked on the development of therapeutic strategies that target the specific mutations driving individual patient tumors, and also on understanding molecular determinants of response or resistance to anti-cancer therapies. A major focus of his work that I think we'll hear about today has been on developing strategies to target colorectal tumors that carry mutations in BRAF and KRAS oncogenes. In addition to his scientific accomplishments, he has been instrumental at MGH in setting up a center-wide infrastructure that has been emulated in other places to systematically integrate important translational facets like specimen collection for serial tumor biopsies, circulating tumor DNA analysis, and patient-derived tumor models into clinical trial design to better understand both sensitivity and resistance to therapy. In addition to several leadership roles inside Harvard, he also is a member of the NCI Colorectal Cancer Task Force and a PI on the Stand Up to Cancer Colorectal Cancer Dream Team. A couple of reminders as Ryan gets started, please leave your microphones on mute. Also, please enter questions during the seminar into the Q&A box of Zoom so we can retrieve them at the end for Ryan to answer. So with that, please join me in welcoming Ryan back to Stanford. Ryan, it's all yours.
So thanks so much, Raj. So great to join everyone today. Good morning. Hopefully we're nearing the time when we can start to do these in person again, but nonetheless, it's great to be talking to you today. So as Raj mentioned, I'm going to be focusing the talk today on overcoming resistance to RAS-MAPK kinase pathway inhibition. And these are my financial disclosures. So despite some marquee successes of precision oncology strategies or therapies that target the specific mutations driving individual patients' tumors, inevitably, even some of the best targeted therapies are limited by therapeutic resistance. And when we approach strategies to understand and eventually to overcome therapeutic resistance, we divide resistance into two broad categories typically. One is primary or upfront resistance, sometimes called de novo resistance. And this is when patients' cancers, despite harboring the target oncogene, are simply resistant to therapy from the get-go. And even with some of the marquee precision oncology successes shown here from the last decade or so, there's typically always a percentage of patients, sometimes about a quarter to a third of patients, who fail to respond to therapy. And understanding this and how to overcome these upfront resistance mechanisms is typically an important first step in optimizing therapy. But in the patients who do respond, unfortunately, inevitably, these patients will at some point develop what we call secondary or acquired resistance to therapy. And so here, I think this is really become a kind of a landmark of photograph to illustrate acquired resistance over the years. This is a patient who had a phenomenal response to a BRAF inhibitor with BRAF V600 melanoma. And unfortunately, just amazingly, just 8 weeks after this middle picture was taken, this patient's tumor recurred quite broadly and dramatically. And this is unfortunately more of the rule rather than the exception we see here. So a really basic way of thinking about how resistance occurs is that we ideally select highly credentialed targets that are altered and lead to constitutive survival and growth signals to which a cancer cell becomes addicted. And if we select these targets right and we're able to target them effectively, we can actually bind that target and cut off these critical signals. And if the cancer cell is indeed dependent on those signals, this will result in profound cell death. So there are a number of basic mechanisms that are used kind of again and again by cancer cells to overcome these therapies. One is actually quite simple. It's actually alteration of the actual drug target so that the drug can no longer bind, and accordingly signaling goes on unabated and the cell is unaffected by this therapy. More nuanced mechanism of resistance involve things like bypass or escape pathways, where the cell may contain a second alteration in either downstream in the same pathway or in a parallel or redundant pathway that make that cell no longer totally dependent on the original altered signaling pathway. And so now, even though the drug can bind and inhibit the original target, another alteration within the cell can sustain growth and survival. And finally, an even more nuanced but increasingly important mechanism is called adaptive resistance. Here again, the drug can bind and at least at first is able to effectively inhibit the target and the pathway, but then through some complex either feedback adaptations or other adaptations, the cell is able to reactivate that original pathway at least to some extent, or to activate perhaps a compensatory pathway in order to maintain survival. And this is something that we'll talk about more today. So obviously, if you're trying to devise a therapeutic strategy to overcome resistance, really understanding the specific mechanism that's operant in each cancer or in each patient is critical, because as you can see here, each of these different common resistance mechanisms will likely require a very distinct approach to overcome resistance. And kind of our belief over the last few years has been, or last several years rather, is that by understanding these mechanisms of resistance, we can really guide the development of rational approaches to overcome therapeutic resistance. And we'll talk about some examples of these today. So I want to start with focusing primarily on upfront or primary resistance. I'll start by illustrating this in an area that our group has worked for almost probably 10 or more years now. And that is in the roughly 10% of colorectal cancer patients that harbor oncogenic mutations in BRAF, specifically BRAF V600 mutations. This is about 8 to 10% of colorectal cancer patients. That confers a terrible prognosis, probably the worst molecular subtype of colorectal cancer relative to prognosis, relative to outcome. But about 10 years ago, specific BRAF inhibitors entered the clinic, and these had phenomenal results in the roughly half of melanoma patients that harbored the exact same BRAF V600 mutation, with response rates in excess of 50%. So at the time, this was really on the forefront of the precision oncology revolution, and we thought that perhaps the same approach would work just as effectively in other cancers that harbored BRAF V600 mutations like colorectal cancer. But unfortunately, when colorectal cancer patients harboring the exact BRAF mutation were treated with the same BRAF inhibitor, in this case vemurafenib, only a 5% response rate was observed, suggesting that there's markedly different sensitivity to this inhibitor. And I think that this example probably to date remains one of the most striking examples of difference between two tumor types as far as responsiveness in the setting of the same mutation. And so our lab became interested in this at the time and really set out to understand what it was about BRAF mutant colorectal cancers that were different from BRAF mutant melanoma and what really preempted response to these agents. So at the time, the prevailing hypothesis was just that, well, colorectal cancers obviously are just less dependent on MAP kinase signaling. They're just less dependent on BRAF V600, and potentially there are other important parallel, perhaps MAP kinase independent pathways, such as the Wnt beta-catenin pathway that's mutated in all colorectal cancers, maybe contributions from pathways like the PI3 kinase AKT pathway that just allow these cancers to survive without signaling, without intact BRAF signaling. So to test this hypothesis, we actually did some very simple experiments where we obtained models of BRAF V600 melanoma and BRAF V600 colorectal cancer, and then looked at the response, the signaling response over time to BRAF inhibitors, in this case vemurafenib. What you can see in BRAF V600 melanoma models is that a BRAF inhibitor will turn off pathway output measured by assessing levels of phosphorylated ERK, and it maintains suppression of that pathway out beyond 48 hours. However, in colorectal cancer models, same mutation, same inhibitor, we see an effective initial suppression of the pathway, but then the pathway comes back on very rapidly and quite robustly. So that on average, by 48 hours, the pathway is at about 50% or more of the basal levels. And this is not due to drug stability or drug efflux. If you replenish stores of the drug, that this will continue. But what this suggests to us is perhaps an alternative hypothesis that in colorectal cancers, at least, these BRAF inhibitors are not able to effectively sustain suppression of MAP kinase signaling. And that this, of course, if you're not inhibiting the target pathway for more than 24 or 48 hours, this is likely a key mechanism that could be driving the lack of initial response. And so to summarize a lot of work that we won't have time to go into in detail today, this really became one of the first examples of adaptive feedback resistance. And mechanistically, the way this works is that when there's an activating alteration within the RAS-MAP kinase pathway, in this case, BRAF V600, this leads to constitutive activation of this pathway, which culminates in activation of ERK. And ERK leads to the transcription of a number of very robust negative feedback signals, such as DUSP and Sprouty genes, which actually disrupt and abrogate activation of this pathway through some of the normal mechanisms, namely the way this pathway is activated in normal cells through signaling, growth factor signaling through receptor tyrosine kinases. However, after a few hours of blocking this signal, you lose these negative feedback signals, which are no longer produced transcriptionally. And now receptor tyrosine kinases like EGFR and others can engage wild-type RAS and essentially reactivate the pathway, bypassing the BRAF inhibitor in this case. So this obviously suggests that if this is the key mechanism that's turning the pathway on, that perhaps by blocking this feedback loop at other points, either by targeting a specific receptor tyrosine kinase that may be the dominant driver of this adaptation, or targeting downstream to maintain pathway inhibition, we might be able to improve outcomes. And so what we found is that the reason that this leads to differential responsiveness in colorectal cancer versus other cancers such as melanoma, and as we've learned over the years, even in other cancers like lung and thyroid, is that colorectal cancers express incredibly high levels of total receptor tyrosine kinases like EGFR, as well as activated EGFR relative to their melanoma counterparts. This is the case both in models as well as in human tumor samples, where you can see the vast majority of these colorectal cancer models, but importantly not all, express very high levels of activated EGFR. So what we did just to set out is if we could target a key RTK like EGFR that might be driving this feedback, can we actually improve the degree of pathway inhibition and the length of pathway inhibition to enhance outcome? And so what you see here in an example colorectal cancer model is by 48 hours with a BRAF inhibitor alone, you're seeing incomplete inhibition of phospho-ERK, and you're seeing a compensatory increase in upstream activity driven by RAS activation and activation of wild-type RAF members. But if you now block BRAF with an upstream inhibitor of EGFR, you now at 48 hours can get a sustained suppression of phospho-ERK, and you block this compensatory adaptive increase in upstream signaling. And then if you look at what happens from an efficacy perspective in xenograft models, either a BRAF inhibitor alone in red or an EGFR inhibitor alone in green has very minimal effect. But when you combine these agents, you now see tumor regressions in the majority of models, suggesting that targeting a feedback loop like this in order to achieve more sustained pathway suppression could be a potential interesting therapeutic strategy. And so this led us and others to lead an iterative series of clinical trials looking at BRAF inhibitor combinations with the goal of suppressing and increasing the sustained nature of pathway inhibition. So initially, we explored a combination of BRAF and MEK inhibition together. And even though this was a downstream block, what we found is that in paired biopsies from these trials, that upstream signaling was able to push its way through this block of downstream blockade of BRAF and MEK alone, leading to pathway rebound. And this led to a modest increase in activity from 5% to 12%, with some patients doing quite well, but really overall was disappointing. We then, based on the data I just showed you, decided to try to target EGFR either alone or in combination with a downstream inhibitor in order to block both EGFR dependent and EGFR independent modes of feedback. And what we found here, again, was a modest increase in activity. But we found actually with triple inhibition an even more great shift in response rate. Again, supporting the hypothesis that if we can block this rapid adaptation and sustain pathway inhibition, we might be able to improve outcome. And last year, a strategy based on this concept called the Beacon regimen, which is a combination of a BRAF inhibitor encorafenib with the EGFR antibody cetuximab, was actually FDA approved based on this randomized study. And this is now the first FDA approved strategy for BRAF V600 colorectal cancer patients. But as we'll get back to toward the end of the talk, there are some remaining issues. So first of all, the response rate, despite having two agents, remains low. Only about 1 in 5 patients have a true response. And the durability, perhaps even more problematic, is quite limited, with a median progression-free survival of only 4 months. So what could be driving these issues? Well, one thing is we actually built into each of these iterative trials paired biopsies at baseline and at day 15, which can actually allow us to assess the degree of pathway inhibition we were achieving with each strategy. And what you can see here is with the BRAF-MEK combination, we were inhibiting the pathway in everybody, but to a very limited degree, probably on average by only about 30 to 40% in patients. And this is compared to what we're achieving just with one drug, a BRAF inhibitor alone in melanoma, where we're getting an average of about 80 to 90% suppression. So this is probably an illustration of this kind of this feedback adaptation by 2 weeks that turns the pathway back on. Interestingly, with BRAF EGFR, what we see is a little bit more of a disparate outcome for different patients, where some patients have very high level suppression. These are likely the patients who do quite well. And in other patients, very minimal inhibition. And in some patients, you actually see what you can see sometimes with BRAF inhibitors is a paradoxical activation of the pathway when upstream RAS activity is driven by a source that is not being inhibited. And finally, with the triple combination, which at least in our series of trials gave the best activity, we're now seeing on average more high-level suppression, approximately 60% inhibition in patients. But amazingly, even with these 3 drugs, we're still falling short of what we're achieving just with a single drug in melanoma. And amazingly, after all this work in 10 years, still our prevailing hypothesis is that our failure to inhibit this pathway more completely is still what's driving the lack of or the limited efficacy with some of our therapies. So I want to just stop and summarize this part of the talk to date before we move on to the next topic. So targeting adaptive feedback can lead to enhanced therapeutic efficacy in BRAF V600, and I think there's both now preclinical and strong clinical data for this. But I think as we're learning, unfortunately, and this is a theme we'll come back to later in the talk, is that while EGFR is a specific RTK, which is quite easy to target, while it may play an important role in many patients, it appears to be critical only in a subset of patients, and that multiple RTKs perhaps are able to lead to this adaptive feedback, which makes our job much tougher. So single RTK strategies may be suboptimal to block adaptive resistance in this setting and in others. And strategies to inhibit EGFR-dependent mechanisms of adaptive feedback really may be key. I think there still is some promise with downstream pathway inhibition. It's perhaps possible that the MEK inhibitors we've used in our trials really are not the best way to do this, since they're very susceptible to upstream override. But we're working on this as we speak. And interestingly, I think some of these same concepts are now rearing their heads again about 10 years later, as we're seeing a very exciting new group of agents now enter the clinic. And so KRAS mutations in particular are the most common oncogenic mutations in all cancers, mutated in about 20% of all human cancer, about 40% of colorectal cancer, 90% of pancreatic cancer, 30% of lung cancer. So probably one of the most important potential targets in oncology. But up until recently, despite about 3 decades worth of efforts, the academic and those in academia and industry have really failed to develop a tractable way to target RAS directly. And this is due to the fact that that RAS is a small molecule GTPase, not a typical kinase that like what we're used to designing precision oncology strategies against. And it really doesn't have a canonical binding pocket that can be exploited. But a breakthrough came about 7 or 8 years ago now from the work of Keyvan Shokat at UCSF, who had a hypothesis that one specific mutation of KRAS, which involves conversion of glycine 12 to a cysteine, or the G12C mutation, presented an opportunity to actually exploit that mutant cysteine present only in the mutant KRAS, and thus only in cancer cells, to actually design molecules that could have undergo a covalent or irreversible interaction with that reactive thiol that's part of the cysteine residue. And by doing this, they were able to screen fragments and develop molecules that actually could bind to a kind of an occult pocket on KRAS that was not previously appreciated. And through further optimization, this has led to a host of inhibitors that have entered the clinic in the last couple of years, initially targeting the KRAS G12C mutation specifically, with some inhibitors shortly behind that look like that they've been able to exploit this pocket to now target other different mutant forms of RAS or RAS more broadly. And initial clinical efforts in this area have shown some tremendous promise. Two of these agents that are furthest along in clinic have shown response rates of about 40% in non-small cell lung cancer. One of these, a drug called sotorasib from Amgen, was just FDA approved for non-small cell lung cancer harboring KRAS G12C mutations about a month or two ago. But interestingly, as this initial clinical data was coming out, we noticed a pattern that we remembered seeing several years ago, where colorectal cancer patients harboring the same mutation were not doing as well. And what we're seeing clinically right now is the response rates in these agents are in the high single digits or low teens relative to response rates of about 40% in non-small cell lung cancer. What we're also seeing in non-small cell lung cancer is that, again, not every patient's responding. Less than half of patients are responding, and many of these patients, their responses are short-lived. So again, this smelled to us very much like our old friend of adaptive resistance coming back to wreak havoc. And so a very talented postdoc in my group, Megan Ryan, set out to really understand what was going on here. And she assembled a large pool of established and patient-derived KRAS G12C models spanning different tumor types. And what she found is actually interesting across the board in this larger panel of approximately 12 or more models, that regardless of the different KRAS G12C inhibitor given, that again, over the course of 48 to 72 hours, you see very rapid and robust reactivation of pathway signaling, such that on average, with all three of these inhibitors, this top inhibitor is now sotorasib, an FDA-approved agent. This inhibitor from Mirati, I think many of us expect may be FDA approved later this year or early next year. Even with these very effective clinical inhibitors, we're seeing pathway rebound back to about 75% of baseline levels by 72 hours. This makes it very tough to expect that we're going to see robust and sustained responses if the pathway is turning itself back on like this. Again, this is not due to an issue with drug stability. If we replenish this drug at regular intervals, we see the same thing. So what's happening here? Well, it's as we've been able to sort out, it looks like this is really the exact same mechanism that we found about 10 years ago in BRAF mutant colorectal cancer. But in this case, MAP kinase signaling is being driven by mutant KRAS. This is leading to the expression of negative feedback signals. And when you inhibit KRAS G12C, this leads to a transient inhibition of the pathway, loss of negative feedbacks, and allows two potential mechanisms to reactivate the pathway. And here's where it gets a little more complicated and nuanced than in the BRAF scenario. So one of these mechanisms actually is that of all KRAS mutations, KRAS G12C actually cycles much more actively between its active GTP-bound state and its inactive GDP-bound state. And it turns out most of the, and if not all of the clinical inhibitors currently are specific for the inactive GDP-bound state. And work from Neil Rosen and Pierre Lito's lab has shown that if you can push G12C more and more into its active state, and one way to do this is by increasing upstream activating input into KRAS, it makes it more difficult for these KRAS inhibitors to bind, and they're less able to inhibit the target and the pathway. So that certainly is one mechanism by which input from RTK can drive resistance to these inhibitors. But a second mechanism is much more similar to what we see in the BRAF setting, where these receptor tyrosine kinases can bypass KRAS G12 altogether by activating the wild-type RAS, wild-type NRAS and HRAS that's present in every cell, and in some cells, wild-type KRAS is retained that are there and capable of being activated by upstream signals. And this can just bypass the inhibitor altogether, turning the pathway back on. Now, why does this matter? Well, this matters because there are some who hypothesize that if we can design a next generation G12C inhibitor that's capable of inhibiting both the active or inactive state of the inhibitors, that this could potentially overcome this resistance if mechanism number one is the primary driver. But if mechanism number two is at least partially or perhaps predominantly responsible for resistance, this is going to bypass G12C entirely, and a new or enhanced G12C inhibitor may not fix the problem here. And so Megan really set out to kind of dissect the contributions of each of these particular mechanisms. And interestingly, what she saw here in this just a single example, I'll show some quantitative and integrated data in just a second, is she found that at an early time point of 4 hours and a later time point at 48 hours, you really don't see a striking rebound in the levels of activated KRAS, or in this case termed KRAS GTP. Yet you're seeing this profound reactivation of the pathway by 48 hours. But what we do see is a very robust activation or increase in the levels of active GTP-bound NRAS and HRAS, the wild-type RAS that are present in all these cells by 48 hours, suggesting that this mechanism number two involving wild-type RAS could be a major contributor to pathway reactivation. And just to show that this isn't just a one-off example, this is actually now integrated across the whole panel. And again, if you look just at activated KRAS levels, KRAS GTP levels, between 4 and 48 hours, even at very high doses, you don't see a clear rebound in KRAS activity, suggesting that reactivation of KRAS may not be having a large effect on driving pathway reactivation. But what you do see is very consistently, approximately a 5-fold on average activation of NRAS and HRAS, suggesting that wild-type RAS signaling bypassing G12C altogether could be a major player in this process. And so if that's the case, developing inhibitors that inhibit the active state of G12C certainly may have some potential, but they may not be able to completely block this second mechanism, which bypasses G12C altogether. And what we may need to do is to think about either targeting individual receptor tyrosine kinases, but as we saw in the BRAF arena, there isn't always a single RTK that leads to reactivation of signaling in all patients and in all cancer types. And so this can be a little hit or miss. So one strategy is to actually target what we call a convergent signaling node, which actually lies between all RTKs and RAS. And one example of this is SHP2, for which there are several inhibitors that have entered the clinic recently. And there are also some additional targets such as SOS or SOS1 that can also have a role in integrating all RTK signaling and connecting it as a common adapter to RAS signaling. So targeting at this point might allow us a way to more universally intercept RTK signaling. And so again, just some very representative data here, but Megan sent out to look across various models, various tumor types, to see if there was one or a small number of specific receptor tyrosine kinases that were largely responsible for driving this type of adaptive feedback. So what we see here is an example of a lung cancer cell line that's actually quite sensitive to an inhibitor. And you see that the addition of various receptor tyrosine kinase inhibitors really do very little to enhance the activity over G12C alone. In this colorectal cancer model, SW1463, you see that a G12C inhibitor alone has minimal effect, but that it combining with an inhibitor of EGFR can actually lead to profound effect. And in this cell line is highly dependent on EGFR-dependent feedback. But in contrast to this other cell line just below it, a pancreatic cell line, the addition of EGFR does almost nothing to enhance the activity over G12C alone. In contrast, this inhibitor is much more sensitive to FGFR signaling, whereas the colorectal model we just looked at really has no, FGFR doesn't contribute much at all. And you see actually a highly varied pattern like this over a spectrum of models. And this suggests, or at least raises the concern, that going after individual receptor tyrosine kinases, even though that might be perhaps easier as a starting point, may not give us the universal efficacy we're looking for. And that's illustrated a little more in detail here, where again, we have a colorectal cancer model that largely appears to be EGFR dependent, at least in vitro, and a pancreatic model that our in vitro data suggested was more FGFR dependent. And what we've done is to treat these either with a KRAS G12C inhibitor alone in red, or combinations with, in this case, a pan-HER inhibitor, which hits EGFR and other HER proteins in dark green, or an FGFR inhibitor in orange. what you can see in this EGFR dependent model is the addition of an FGFR inhibitor does almost nothing relative to a G12C inhibitor alone. And initially, there does seem to be some benefit of combined targeting of EGFR, but this effect seems to be lost over time, such that by these later time points, there's no statistical difference. In contrast, in this model, it's more FGFR dependent. The addition of an EGFR pan-HER inhibitor does nothing relative to a G12C inhibitor alone. And if anything, again, there's a trend towards some benefit to an FGFR combination, although this doesn't quite reach statistical significance. If you look at a signaling perspective at total RAS output, you see that again in this model, an EGFR combination improves pathway inhibition over a G12C inhibitor alone, whereas FGFR does not. And we have the opposite pattern in the more FGFR dependent model. But notably, targeting a convergent signaling node like SHP2 here show that you're able to effectively inhibit this pathway signaling in both models. And this really suggests that coming in at a convergent node might help us overcome this problem of RTK promiscuity. And this is just, again, integrated data over the entire panel here showing that by adding a SHP2 inhibitor, we're able to achieve much more long-term and sustained suppression of the pathway across all models. Notably, what we're seeing here is again, not so much a difference in the level of KRAS, activated KRAS at this 48-hour time point, but rather we're blocking this compensatory increase in wild-type RAS signaling with the combination of SHP2 and G12C. Now, while we're not seeing any difference in KRAS rebound, what we do see, in fairness, is some improved suppression of KRAS initially. This actually may be relevant more toward the fact that these inhibitors are able to engage and inhibit more rapidly the inactive state of KRAS. So this is where perhaps an active state KRAS inhibitor might have an advantage, though it may not be enough to overcome the full rebound through wild-type RAS. So does this theory pan out in vivo? Well, now we've taken these same two models that had differing reliances on EGFR and FGFR. And what we found is that actually with a SHP2 inhibitor, we're actually able to achieve tumor regressions in both of these models and achieve enhanced and longer-term suppression of the pathway. So this has been shown by other groups, including data here from a group at Mirati. The big question now, though, is that SHP2 inhibitors are rather new to the clinic, and obviously inhibiting RTK signaling in general certainly has the potential to lead to some adverse events. And so the big question is whether these agents will be tolerated well enough in clinic to support combinations of this nature. So with that, I think there really are a couple key strategies that we're trying to take forward in the clinic right now to target RAS. I think really job number one is focused on enhanced pathway targeting. So we discussed the rationale for upstream targeting. There are strategies mimicking BRAF in colorectal where we're targeting EGFR specifically with G12C inhibitors in colorectal cancers, and there are ongoing strategies with SHP2 inhibitors and other potential upstream points. There's also thought of involving downstream inhibitors, either alone or in combinations with some of these upstream agents. And I really argue that until we're able to sustain suppression of this pathway, this is really job number one. But once we are able to do that, thinking about actually now enhancing the activity of these agents by combining with things outside the pathways. There's been proposals of targeting CDK4/6, some of the anti-apoptotic machinery, or even potential cooperation with immunotherapy, which is a concept we'll come back to in just a moment, could be a way to go. So with that, I want to transition. We've talked mostly about overcoming adaptive primary resistance, but when these patients do respond, what do we do about the eventual emergence of acquired resistance? And what I'll summarize here is just some data that we just recently published a couple months ago. It's one of the first reports of clinical acquired resistance to these new KRAS G12C inhibitors. The work of another postdoc in my lab, Noritaka Tanaka, as well as some collaborators of mine, Becca Heiss and Jess Lin in the thoracic oncology group at MGH. So this was actually a very notable initial patient who went on to have a very nice initial response to the Mirati G12C inhibitor, but unfortunately developed disease progression fairly rapidly after about 4 to 5 months on therapy. As we do with a lot of our patients on interesting trials, is we follow these patients with serial biopsies and importantly with serial plasma draws every 1 to 2 months through therapy so that we can do a comprehensive analysis of circulating tumor DNA to try to identify potential mechanisms of resistance that emerge during therapy. And that's what we saw here. So prior to treatment, both in the baseline tumor sample and in the baseline cell-free DNA, we saw the presence of a KRAS G12C mutation as well as a clonal P53 mutation, but nothing else were seen in these samples. However, after the patient progressed on this KRAS G12C inhibitor, we saw the emergence of a multitude of about 10 or 11 distinct potential candidate resistance mechanisms to the KRAS G12C inhibitors that we explored in more detail. And this really painted a very interesting pattern. It looked incredibly complicated at first, but very rapidly became clear that all of these alterations really clustered within the RAS-MAPK pathway. And we saw about 4 different classes of alterations affecting RAS itself. Some of them involved mutation of a different RAS altogether, in this case, NRAS. And obviously, if you activate NRAS, this cell will perhaps no longer be dependent on KRAS and thus no longer dependent on a KRAS G12C inhibitor. We see somewhat similar in nature alterations in KRAS as well, where what we're either seeing is either a loss of G12C, where it gets turned to a different KRAS mutation that no longer can support this irreversible interaction, or perhaps more likely these mutations are occurring in trans, where the remaining wild type copy of KRAS, which was retained in this patient, is actually mutated, so it can no longer be, it can provide KRAS activation that's no longer inhibitable with a KRAS G12C inhibitor. And then interestingly, we did see a very small number of reads, but what did give some support for a G12F mutation, which is interesting because it actually suggests kind of a 2-stage mutation originating from the original KRAS G12C mutation with a second nucleotide mutation that leads to G12F. G12F is very rare because it actually takes 2 nucleotide changes to get there from the wild type copy, suggesting that this likely occurred in cis. We also saw, I'll get to the secondary KRAS mutation in general. This is something that I'll talk about in just a moment. But we also saw a number of downstream mutations in BRAF and in the MEK genes, which suggest that at least reactivation of the MAP kinase signaling pathway only, which is not the only pathway that KRAS turns on, could be sufficient for blocking activity. One of the more interesting things that popped out was this mutation, this KRAS Y96D. So interestingly, if we searched hundreds of thousands of patients in the Gini and Sanger databases, not a single mutation at Y96 was identified, despite KRAS being the most commonly activated or mutated oncogene in cancer. And I think before I get into the mechanism of KRAS Y96D specifically, I think this raises an important point that is a big challenge that we face in the setting of acquired resistance. And this is the development of sometimes extensive tumor heterogeneity upon acquired resistance. This can either be intralesional, where different metastatic lesions can independently evolve different resistance mechanisms, or it can be intralesional, where actually different subclones residing within the same lesion can actually independently involve resistance. And this has really been a challenge for us trying to identify what's driving resistance, but also figuring out how to target resistance in a patient. If we were to biopsy just one of these lesions, we might have just identified perhaps just one of the alterations that were present in this patient. And this is really where cell-free DNA has given a huge advantage, because circulating tumor DNA is shed by tumor cells throughout the body and can give us a much more complete catalog of what's going on. And when we actually looked at this in more detail in a panel of GI cancer patients we published a couple of years ago, we found that if you look at actually matched post-progression tumor biopsies following targeted therapy relative to matched post-progression cell-free DNA, that a couple things are evident. Number one is we're much more effective at developing or identifying at least one resistance alteration using cell-free DNA. But we also identify a lot more patients, as shown by this orange piece of the pie, who have multiple resistance alterations. And I think one of the key findings was we actually looked at the patients who had time-matched post-progression tumor biopsies and post-progression cell-free DNA. We actually identified clinically relevant laboratory-validated resistance mechanisms not present in the tumor biopsy in about 80% of the time. So we've really moved to rely on liquid biopsy for understanding the heterogeneity of acquired resistance for reasons just such as this. So while a lot of these mechanisms represented simple bypass tracks, we really focused on this new mutation. And what we found is that this actually lies within the switch II binding pocket of KRAS, and actually has a direct interaction with all three of the the model early inhibitors. Different interactions, but important actions nonetheless. And when this residue is mutated, this disrupts and affects binding of these inhibitors. And what we found is that this mutation was able to universally inhibit activity of these various inhibitors, and would lead to the inability of these inhibitors to turn off KRAS signaling and maintain RAS-MAPK kinase pathway suppression. So because this had a direct interaction with this class of inactive state specific inhibitors, we asked, are there structurally unrelated KRAS inhibitors that might actually have an advantage in overcoming these secondary mutations, which we are starting to see more and more, both in our own data set as well as in another data set published by our colleagues at Mirati and Dana-Farber just recently. Now, there's a new class of inhibitors called Tricomplex inhibitors that actually are specific to the active state of KRAS. And this is a very interesting mechanism. These actually use the same mechanism as drugs such as cyclosporin or rapamycin, where they actually bind to a chaperone protein, in this case, cyclophilin A, the same chaperone is used by cyclosporin. And then one part of the molecule will associate with the chaperone, the other part will initiate a specific interaction with a target. But then this large kind of amorphous protein surface of the chaperone can then is quite malleable and can actually form a number of sometimes lower affinity, but multiple interactions, almost like Velcro with the surface of this target, and can enhance the avidity of that interaction, allowing the ability to target harder to drug surfaces. So this specific inhibitor, RM18, is an early example of this. And we actually asked if this inhibitor could overcome this. And what we see here is that this inhibitor is actually no longer affected by this Y96D mutation. And interestingly, is able to sustain pathway. It's the only inhibitor that can now sustain pathway suppression in the setting of this Y96D mutation. The other interesting finding is that since all these inhibitors have a covalent interaction with G12C, you can see this kind of band shift in KRAS, where you get a higher molecular weight band that's the kind of irreversibly adducted protein. You lose that upper band in the setting of the Y96D, suggesting that binding is disrupted, with the exception of the RM18 tricomplex inhibitor. So this really leads us to kind of a quandary here, is that how are we going to come after a KRAS if we have to deal both with this upstream adaptive resistance and the emergence of actually a number of downstream resistance alterations? We can certainly try to block adaptive resistance upstream, but this is not going to block many of the acquired resistance mechanisms we saw. And so there's now kind of increased interest in either bringing in downstream inhibitors or at least perhaps increasing the importance of these downstream inhibitors in our strategy. So again, to summarize here, combinations therapies for KRAS cancers remain a promising approach. Adaptive feedback reactivation, much like in the BRAF setting, can be a key limitation when targeting the pathway. And targeting convergent signaling nodes may provide advantages and more universal efficacy. Acquired resistance appears to emerge due to multiple polyclonal RAS-MAPK kinase alterations leading to pathway reactivation, and novel RAS inhibitors and/or downstream inhibitors may be needed. And so I want to finish just with a few slides to really raise the questions, is as we're struggling to overcome this kind of polyclonal heterogeneous resistance, is targeted therapy or targeted therapy combinations ever going to be enough? And it's looking like some of these resistance pictures become so complicated that it could just be too difficult to design a targeted therapy combination that's tolerable, that will also allow us to completely overcome the full spectrum of resistance. And so this has led us to think about kind of integrating targeted therapy approaches with other modalities. And to shift gears back to BRAF V600 colorectal cancer, some data from this came as we look back to our trials of targeted therapy only in BRAF V600. One of the notable things about BRAF V600 is there's a substantial overlap with microsatellite instability in colorectal cancer. And as many of you may know, microsatellite unstable cancers have higher mutational burdens and respond much better, have a more avid immune response, and respond well to immunotherapies. So if we look back at our trials, again, just targeting BRAF, we noticed something interesting, that those patients who also harbored MSI, and a subset of those patients, they went on to have very good and very durable responses. In fact, any patient who was on for more than a year happened to be MSI. And there's one really remarkable patient from our very first trial of BRAF-MEK, who's probably about 8 or 9 years out now with a complete response off of therapy. Again, nothing notable about this patient's tumor other than it was also MSI. So this suggested that there may be some interaction between targeting BRAF and the immune response. And when we actually went back and looked at our pretreatment and day 15 biopsies from, again, our targeted only trials, we found that engaging the BRAF pathway with BRAF EGFR or BRAF EGFR MEK led to actually an increase in T cell signatures consistent with increased T cell infiltration, as well as cytotoxic T cell signatures, as well as a number of other potential immune populations. We modeled this further using some immune competent syngeneic BRAF V600 colorectal models in the mouse and found that there appeared to be synergy between blocking immune checkpoint with, in this case, BRAF MEK inhibition. And this led us to initiate a clinical trial about 2 years ago using a BRAF and a MEK inhibitor with a PD-1 inhibitor based on some safety data existing from melanoma patients. Now, while BRAF MEK is certainly not the optimal way to target the pathway, and I'd argue this approach might work better if we used a more effective core, what we do know is that the response rate to BRAF MEK was about 7%, confirmed response rate in patients, and the response rate, at least in microsatellite stable patients to immunotherapy alone, is about 0%. And at least based on this data cut from last year, what we're seeing now is actually about a 33% response rate. So well above not just what we'd expect with these individual components, but about between about 50 to 100% greater than what we're seeing with the current FDA approved standard. And importantly, we're seeing that this is not limited to the occasional MSI patient, that is actually quite effective in MSS patients. So to understand what's actually perhaps leading to this, not just higher response rate, but increased durability that we're seeing with some of these agents, we've, which is shown here, our best patient was on for about 2.5 years. We've actually now integrated again these baseline and day 15 biopsies, but actually are now doing single cell RNA sequencing from these populations. And while technically challenging, this has allowed us to look at some really interesting questions, looking not only at cell changes here, you see a very marked influx of T cells in this complete responding patient, a decrease in tumor cells, but we can kind of hone in on these individual populations and look at the gene expression changes. And so specifically looking at tumor cells, we can see that we're at least getting some degree, or perhaps somewhat incomplete, but some degree of pathway suppression of the MAP kinase signaling pathway. And in the T cell population, in addition to having increased numbers, we can see that we're seeing altered expression, in this case, increased expression of cytotoxic targets. What it's also allowed us to do is to dig in and try to ask what's happening differently in our patients who are responding and non-responding. And what's emerged very interestingly is that we've seen that responding patients seem to have marked induction of several important immune modulatory pathways, including interferon response pathways and antigen presenting pathways, again, entirely within the tumor cell. What's notable is when we take patient-derived organoids from these same patients, we try to generate these from all the baseline biopsies of these patients, and we treat them isolated from the tumor microenvironment with the BRAF and MEK combination. We see that those organoids that come from responding patients have the same induction of these key pathways, and those that come from the non-responding patients do not. And so what this is really suggesting is there's some potential key mechanistic links of programs regulated downstream of the MAP kinase pathway that may actually enhance the interaction with the immune response. And this is something that I think will be relevant both in the BRAF setting as well as in the KRAS inhibitor setting as well. So with that, I'd like to end, and I hope what we've seen today is some examples of how understanding resistance mechanisms may guide strategies to overcome resistance. I think distinct resistance alterations arise between patients and within an individual patient, really requiring us to think about this. Liquid biopsy really does better capture the heterogeneity of resistance, which can be missed by single lesion tumor biopsies. And diverse resistance mechanisms are very difficult to target, but as much as possible, we need to identify where these converge on key signaling nodes. And our combination strategies should perhaps focus on these convergent nodes to allow more universal strategies. Upfront targeting of these convergent signaling nodes may prevent outgrowth of these pre-existing resistant subclones, and ultimately integrating other therapeutic modalities such as immunotherapy, even cytotoxic therapy, may be critical to eradicate these resistant subclones for more durable responses and or cure. So with that, I want to end here and thank those who contributed this work, as well as my funding sources. And with that, I'd be happy to take any questions. Thank you so much for your attention.
Thank you, Ryan, for that really systematic analysis of the RAS-MAP kinase pathway in cancer and for giving us a sobering view of how difficult it is to inhibit this pathway despite multiple therapies we have. I want to encourage people in the audience to put their questions into the Q&A box. And while we wait for questions to pop up, I'll start with a couple. I want to go back to your observation that the feedback mechanism seems to be much more important in colorectal cancer versus melanoma, and you mentioned that might be due to overexpression of EGFR. Is it, I'm wondering why the pathway through these other receptor tyrosine kinases is more important in colorectal cancer? Is there something about the expression sort of profile of receptor tyrosine kinases? Is it sort of a lineage thing, or because you would think that melanoma cells should also be able to upregulate other receptor tyrosine kinases to bypass RAF inhibition?
Yeah, no, this is a great question, and I think it's not as simplistic as that one slide suggested. I think, but I think there could be lineage effects as well. Obviously, melanoma is not an epithelial cancer and derived more from neural crest cells. So there could be some more lineage specific effect there, but you actually see a very consistent pattern even across epithelial tumors. So if you kind of start with the BRAF V600 where there's the most data, single agent BRAF inhibitors yield response rates of about 50 to 60% in melanoma. In non-small cell lung cancer and thyroid cancer, they produce response rates of about maybe 30 to 40%. And then in colorectal cancer, 5 to 10% at best. Now that same pattern, we're seeing that kind of play out in the KRAS G12C setting, where again, lung cancer, we're seeing about a 35, 40% response rate with some of these initial inhibitors and kind of a single digit to low teens response in colorectal cancer. So it really does seem to be hardwired in. I think part of it could be the raw expression of RTKs. Part of it could be the microenvironment and the kind of the abundance of certain RTK ligands. But what we're finding is that even in colorectal cancer, where a lot of focus has been kind of targeted on EGFR, we're seeing a lot of other receptor tyrosine kinases can kind of step up and promote the signal. So I think the short answer is we don't know, but it paints a very complicated picture where I think we may see some success. Like I do predict that the EGFR G12C combinations, for example, will certainly, I believe they will enhance activity in colorectal cancer. We'll hopefully know soon. But I think just like in BRAF, we're going to hit a little bit of a ceiling where we're not going to kind of get to these 60, 70% level response rate levels that we want to until we can inhibit more broadly.
I see. We have a couple of questions. One from Jim Ford. And Jim asks, so does BRAF MEK inhibition induce MSI and immuno-oncology responsiveness in colorectal cancer?
Yeah, so great question. We actually don't think that it induces MSI. We really don't see a marked change in the kind of the mutational profile or the mutational burden of these cancers. Colorectal cancer actually isn't, it's not necessarily a quote unquote low mutational burden tumor. But for its mutational burden, it responds much more poorly. So we really do think that there are some additional mechanisms beyond that kind of constrain the immune response. So what we are seeing very consistently now, not just in patients, but in our models, is that two things are happening that are different in the responders and the non-responders. And I touched on this briefly, that we see these kind of marked immune programs go up in the tumor cells only. And we can recapitulate that in standard cell lines, patient-derived organoids. So it really does seem to be hardwired into the pathway. There seem to be chemokine programs, interferon response programs, innate immunity programs, and even antigen presenting programs where we see upregulation of MHC. So it almost suggests that evolutionarily that the MAP kinase pathway seems to suppress the immune response. And if you think about this as largely a wound healing pathway, it may make sense that there could be some direct hardwired programs that prevent kind of a self-immune response that derive from that. The other thing that we see in non-responders is we see kind of a failure to suppress the MAP kinase pathway. So back to our original problem. And the interesting thing is that could be a chicken and the egg, or it could be true and unrelated. But because we have these models, we can now go there and we can take kind of better MAP kinase alterations, strategies better than BRAF MEK, such things like MEK-ERK, which can more universally inhibit the pathway. And when we do that and can get high-level pathway inhibition, we now see these immune programs go up across the board in these patients. And so we actually think that this is hardwired into a larger percentage of these patients. And if we can achieve higher level pathway suppression, we can actually perhaps kind of promote this immune response. So we're still learning about this, but that's kind of our working model right now.
Right, we have another question from Steve Artandi. Steve writes, thank you for the great talk and for presenting your beautiful work on adaptive responses in the RAS pathway. His question is, are there any differences in how these cells respond to CRISPR-based elimination of BRAF or RAS, for example, compared to small molecule inhibition of these oncogenes? So is the adaptive response different between genetic knockout versus small molecule inhibition?
Yeah, so this is a great question. And this even goes back, before we had CRISPR where kind of knockdown, there was a lot of papers in around 2009, 2010 that would knock down mutant KRAS with shRNA and show that some cell lines were kind of KRAS dependent versus KRAS independent. Now, you can argue the degree of knockdown might not have been perfect, but we are kind of seeing this again, where you can actually completely ablate some of these oncogenes, and amazingly enough, feedback through the wild type pathway can actually support continued growth of maybe not every single cell, but at least a substantial subset of those cells. So it does suggest that that even if you could kind of completely ablate the oncogene with pharmacologically or through some other method, that you might not totally kind of overcome this issue.
Great, thank you. One follow-up question I had on that, Ryan, was, are there any efforts to, it seems like part of the problem is this feedback that gets augmented when you inhibit upstream. Are there pharmacological strategies to augment that feedback in some way?
Mm-hmm. We've tried, other than inhibiting the nodes, one of the thoughts is how do you, can you actually, target some of these feedback mechanisms directly? I think the issue is that what we really need to do is almost kind of force sustained expression of particularly, I think, some of the sprouty proteins, which seem to be the ones that really decouple RTK signaling from RAS. And I don't think we have a clear way to do that. But obviously, if there was a kind of a negative regulator of negative feedback inhibition, we could potentially target that more directly. That's something that there's been discussions about, but I don't think there's a clear path on how to go. I think that was your question. If it wasn't,
yeah, that's it. No, that's right. Yep. All right, and then we have another question from Nathaniel Gray. Any progress with epigenetic or transcriptional targeting combinations with upstream signaling small molecules?
So Nathaniel, how are you? Yeah, so the short answer, probably no. I think this is certainly something that we're thinking about a little bit, and I do think that there's a really interesting. I think we're seeing kind of an emergence of a new area of therapeutics where we can actually target. Not just target the protein state of various genes, but we can actually interfere with either transcription or really, I guess, more translation of gene products at the transcriptional level. That's certainly something we're thinking about. Just I think we don't quite have the tools to go through that. But the epigenetic nature of some of these different, when you look across the different responsiveness of different cancers, it does suggest there certainly could be some type of an epigenetic role in regulating kind of how robust this RTK-driven feedback is. In short, our understanding is probably not deep enough to really act on that. A great question.
All right, so I think we're at the top of the hour. Thank you, Ryan, again, for really a fantastic and clear seminar, for joining us this morning, and thank you all for attending.
Great, thanks everyone. Have a great day.
Bye-bye.